So now complex tissues. Complex tissues means this tissues formed by different kinds of cells, so called complex tissues or heterogeneous tissues. Generally, complex tissues help in conduction, so complex tissues are also called vascular tissues. These vascular tissues first appear in pteridophytes, and plants with vascular tissues placed under Tracheophyta. Tracheophyta includes pteridophytes, then gymnosperms and angiosperms. Under these primitive tracheophytes, pteridophytes, advanced tracheophytes, angiosperms. Rightly, these vascular tissues, it includes xylem and phloem. And these vascular tissues, two types, primary and secondary vascular tissues. Primary vascular tissues, these are primary xylem and primary phloem. These primary vascular tissues are developed from procambium. Then secondary vascular tissues, it includes secondary xylem and secondary phloem. The secondary vascular tissues are developed from vascular cambium. So point, vascular tissues, xylem and phloem, there are two types, primary and secondary vascular tissues. Primary vascular tissues, xylem and phloem, develop from procambium. Secondary vascular tissues, secondary xylem and phloem, develop from vascular cambium. Then the xylem and phloem terms were given by xylem and phloem terms were given by Nageli. Xylem is also called hydrome. Phloem is also called leptome or bast. Here, xylem and terms were given by Nageli. Hydrome and leptome terms were given by Haberland. Next, individual description. First one, xylem. Xylem help in conduction of water and minerals. Now, parts of xylem. Xylem shows trachary elements. Then xylem parenchyma and xylem fibers. These are parts of xylem, trachary elements, xylem parenchyma and xylem fibers. Rightly, conducting cells or conducting elements of xylem are called trachary elements. It includes tracheids and vessels. Tracheids and vessels. So total parts of xylem, tracheids, vessels, xylem parenchyma and xylem fibers. Complete xylem is dead tissue, but living tissue of xylem, xylem parenchyma. Then first one, tracheids, primitive conducting elements. Primitive conducting elements are called tracheids. Then tracheid is elongated tube-like cell. Tracheid, elongated tube-like cell, middle part is wide, then end walls narrow or tapering. In tracheid, that cell wall is made up of lignin. and a trachea is dead cell without protoplasm. So at center there is empty space. That empty space is called lumen. Generally, tracheids are partially overlapped, and in tracheids that a conduction takes place through bordered pits. And a tracheids present in all tracheophytes, means present in pteridophytes, gymnosperms, and also angiosperms. Main conducting cells in pteridophytes and gymnosperms, tracheids. In angiosperms, along with tracheids, that vessels present in angiosperms. Then next one, vessels. Vessels also called trachea. Vessels, another name for vessel is trachea. And what is vessel means? Vessels are many xylem elements or xylem cells. All these are placed one above the other and forms a row of cells. Then in end walls, perforations are formed. This end wall with the perforations are called perforation plates. Then point, there are two types of perforation plates. One is simple perforation plate, entire end wall has one perforation, or a compound or multiple perforation plate, end wall has many perforations. Then like tracheid, even vessels are dead cells. When vessels dead cells, then without protoplasm, at center empty space is present, rightly called lumen. Because of this empty space, that a flow of water easily takes place from base to top or between vessel cells. So in both the tracheid and vessel, that secondary wall is made up of lignin. But this lignin deposition is of different types. One is this lignin deposition is called annular. Annular means that lignin deposition is ring-like. If lignin deposition ring-like, it is called annular. Then second one, that lignin deposition is a spiral. Spiral means the deposition is spring-like, called a spiral. Then next one is scalariform. Scalariform means that a lignin deposition is ladder-like. If lignin deposition is ladder-like, it is called scalariform. Then next one, reticulate. Reticulate means that lignin deposition is net-like. If lignin deposition net-like, it is called reticulate. Next one, this lignin deposition is in the form of pits. If lignin deposition is in the form of pits, it is called pitted type. These are types of lignin deposition present in both tracheids and vessels. Then here tracheids and vessels, they help in conduction, and their cell wall is made up of lignin. So along with conduction, they also provide mechanical support. Then vessels are characteristic feature of angiosperms. Vessels formed in angiosperms. But in some primitive angiosperms, vessels are absent. Vesselless angiospermic families, Winteracea, Trochodendracea, and Tetracenteraceae. In these families, Tetracenteraceae 𝗧𝗦𝗃𝗃. In these families, the vessels are absent. Then vessels absent in gymnosperms, but some gymnosperms like 𝗧𝗪𝗃 and 𝗏𝗃, in that vessels are present. Not in all 𝗧𝗪𝗃 species. There is one species, 𝗧𝗪𝗃 𝗚𝗏𝗧. In 𝗧𝗪𝗃 𝗚𝗏𝗧, that vessels are present. Generally, vessels absent in pteridophytes, but some pteridophytes like 𝗧laginella and 𝗂𝗽𝗱, they contains vessels. So these are about tracheids and vessels. Next one, xylem parenchyma. The only one living tissue present in xylem is xylem parenchyma. Then the xylem parenchyma is a living cells made up of cellulose, and this parenchyma contains vacuolated cytoplasm, and there are two types of xylem parenchyma. One is axial parenchyma and second one is ray parenchyma. Axial parenchyma. This axial parenchyma arranged in vertical rows along with vessels, and generally primary xylem contains axial parenchyma. Then next one, ray parenchyma. Ray parenchyma means they arranged in radial rows. Radial rows. And a ray parenchyma present in secondary xylem. Actually, function of xylem parenchyma, it help in storage. Like it stores starch and also fats. Along with storage, this parenchyma help in conduction. Particularly, ray parenchyma, it help in radial conduction. Radial conduction means that a lateral conduction. It is xylem parenchyma. Next one, xylem fibers. Xylem fibers, they are sclerenchymatous. Xylem fibers are sclerenchymatous. And xylem fiber is elongated cell. Middle part is wide. End walls narrow or closed. And xylem fibers, they provide only mechanical support. And xylem fibers are two types. One is libriform fibers made up of simple pits, and second one is fiber tracheids. These fiber tracheids made up of bordered pits. So these are parts of xylem, and main function of xylem is that conduction of water and minerals, that too only from base to top, that is called unidirectional, or such flow is called ascent of sap. Xylem is of two types based on origin. One is primary xylem. This primary xylem present from starting stage help in formation of primary parts, and primary xylem is developed from procambium. The next one, secondary xylem. Secondary xylem, also called wood. The secondary xylem is developed from vascular cambium. Developed from vascular cambium. And primary xylem is of two types. One is protoxylem, that is newly formed xylem called protoxylem. Generally, protoxylem contains narrow vessels. Then second one, metaxylem, later formed xylem, it's called a metaxylem. This metaxylem contains broad vessels. Then xylem differentiation. Xylem differentiation is of two types. One is centripetal and second one centrifugal. Centripetal differentiation Centripetal means xylem differentiation is from periphery to center, called centripetal. So in centripetal, protoxylem towards outside, metaxylem towards inside. Such condition is called exarch, and exarch is characteristic feature of roots. And second one, centrifugal differentiation. Centrifugal differentiation. In this type, xylem differentiation is from center to periphery. It means in this type, protoxylem inside, metaxylem outside. Such condition is called endarch. This endarch is characteristic feature of stems. In some primitive plants like ferns, both the centripetal and centrifugal differentiation takes place. Because of centripetal and centrifugal differentiation, the protoxylem is surrounded by metaxylem. Such condition is called mesarch. So these are types of xylem. Next type of complex tissue is called a phloem. Phloem is also called leptome or bast. Then function of phloem, it help in translocation of food from source means photosynthetic part, to sink, that is sink means storage pods. Then parts of phloem, generally phloem shows sieve elements. Then it shows phloem parenchyma, phloem fibers, and companion cells. Point, conducting cells of phloem are called sieve elements. Then sieve elements are two types, sieve cells and sieve tubes. Complete phloem is living tissue. Dead tissue of phloem fibers. And primitive conducting cells, sieve cells, their structure resemble to tracheids. Then these sieve cells present in pteridophytes and gymnosperms. What is sieve cell? Sieve cell is elongated cell. Like tracheid, sieve cell is also elongated cell, and their end walls narrow or closed. Then lateral walls contains sieve pores, and an area with a sieve pores is called sieve area. Then sieve cells are living cells made up of cellulose, and they contains vacuolated cytoplasm. And a matured sieve cell is without nucleus. And these are main conducting cells in pteridophytes and gymnosperms. Next one, sieve tubes. Sieve tubes, structure of sieve tube similar to vessel. Just like vessel, all phloem elements, they placed one above the other. Then their end walls are joined. After that, in end walls, that perforations are formed. These perforations like sieve. Hence, these are called sieve plates. Generally, sieve plates are oblique, and the sieve plates are two types. Simple sieve plate, entire end wall is only one sieve plate, that is called simple sieve plate. Example for simple sieve plate, cucurbita. The next one, multiple or compound sieve plate. involve as many sieve plates called a multiple or compound sieve plate, and example for compound or multiple sieve plate is a 𝗧𝗧. Then like a sieve cell, even sieve tube. The sieve tube is enucleated, matured sieve tube, enucleated, and it contains vacuolated cytoplasm. Then the cytoplasm shows streaming movements or that cytoplasmic movements. And the sieve tubes, they are useful for conduction, particularly the cytoplasmic streaming. They translocate food from one sieve tube to another sieve tube through these sieve areas. Even sieve tube wall is made up of cellulose. Then associated with the sieve tube, one or two or many specialized parenchymatous cells present. The specialized parenchymatous cells are called companion cells. Then sieve tubes and companion cells are connected by primary pit fields. Sieve tubes and these companion cells connected by primary pit fields. Then through these primary pit fields, the cytoplasmic strands are traversed. Then matured sieve tube is enucleated, but matured companion cell, it has nucleus, and the nucleus of this companion cell controls metabolic activities of the sieve tube. That is why sieve tube is enucleated, but it is living. And in sieve tube and companion cells present only in angiosperms. In gymnosperms and pteridophytes, these are absent. In pteridophytes and gymnosperms, these are sieve cells present, particularly in gymnosperms. Along with the sieve cell, that albuminous cell is present in gymnosperms. These are sieve cells and sieve tubes. The next one, phloem parenchyma. Like xylem parenchyma, even phloem parenchyma is of two types. One is axial parenchyma, arranged in vertical rows, present in primary phloem. Then secondary phloem. Secondary phloem contains that ray parenchyma. Ray parenchyma present in secondary phloem. This ray parenchyma, they help in lateral conduction. An important point is phloem parenchyma absent in monocot stems. Then last one, phloem fibers. Phloem fibers, they are dead cells. Then sclerenchymatous. Dead cells, sclerenchymatous. Phloem fibers are also called bast fibers. Then generally phloem fibers are absent in primary phloem, present only in secondary phloem. These phloem fibers are commercially valuable. Like that, some of the examples. This is a flax fiber, it is obtained from linum. Then hemp, it is obtained from that cannabis hemp. Then sun hemp, it is obtained from Crotalaria. Then like xylem, even phloem is of two types, primary phloem and secondary phloem. Primary phloem is formed from procambium. Secondary phloem, it is formed from vascular cambium. Then primary phloem is of two types, protophlloem, that is first formed phloem, metaphlloem. Metaphlloem is later formed phloem, rightly called metaphlloem. And this phloem shows only one type of differentiation. That is what means centripetal differentiation. So this is about phloem, and final conduction of phloem, and finally function of phloem is it help in conduction of food materials. Now next one is a small tabular form, parts of xylem and phloem present in which plants and absent in which plants we can see. Here pteridophytes, gymnosperms and angiosperms. These are tracheophytes. Then tracheids, vessels, sieve cells. Then next one, sieve tubes. The next one, companion cells. And next one, albuminous cells. So these are parts of xylem and phloem. So first one, tracheids, present in pteridophytes. Then tracheids, present even in gymnosperms. Tracheids, present even in angiosperms. It means these are the only conducting cells. Tracheids present in all tracheophytes or all vascular plants. Primitive conducting cells are tracheids. Then lone tracheids present in pteridophytes and gymnosperms. Vessels and tracheids present in angiosperms. Next, vessels absent in pteridophytes. Then vessels absent in gymnosperms, but vessels present in angiosperms. Next one, sieve cells present in pteridophytes. Sieve cells present in gymnosperms, but sieve cells absent in angiosperms. Sieve tubes absent in pteridophytes, absent in gymnosperms, but present in angiosperms. Companion cells absent in pteridophytes, then companion cells absent in gymnosperms, but companion cells present in angiosperms. Next one, albuminous cells, right? Albuminous cells absent in pteridophytes, present in gymnosperms, absent in angiosperms. So this is clear. So these are conducting cells of xylem and phloem. So one small primitive conducting cells in xylem, tracheids. Advanced conducting cells in xylem, vessels. Primitive conducting cells in phloem, sieve cells. Advanced conducting cells in phloem, sieve tubes. Rightly, sieve tube and a companion cell, both are developed from same mother cell. So sieve tube and companion cells are called sister cells. That is why if sieve tube is not formed, companion cell is also not formed. Your vascular cambium, it produces two types of wood. One is heartwood and second one is sapwood. Here vessels of heartwood is blocked by balloon-like structures. These balloon-like structures develop from xylem parenchyma called 𝗘𝗧𝗧. That is why vessels of heartwood not useful for conduction, and sapwood, this newly formed wood, it is useful in conduction. Rightly, these 𝗘𝗧𝗧s are called xylem plugs. Then next one, phloem. In phloem, vessels. In phloem, inside sieve tube, a special protein is present. This special protein is called P protein or slime. P protein or slime, they blocks these sieve plates. So conduction is stopped. Generally, it happened in injured phloem. Then old phloem. In old phloem, callous plugs are formed. Callous plugs means here adjacent sieve tubes connected by cytoplasmic strands. Around that cytoplasmic strands, a thin carbohydrate layer is present. This thin carbohydrate layer is called a callus. And old phloem, this callus blocks that sieve plates, so rightly called callous plugs. This is additional information for xylem and phloem, because last year in EAPMT, these xylem plugs or 𝗘𝗧𝗧 question asked. Next type of permanent tissues, special tissues. Special tissues, also called secretory cells. They produce substance and release substance as a result of metabolism. The special tissues divided into two types. One is external secretory cavities and internal secretory cavities. External secretory cavities, it includes digestive glands, then nectar glands, then 𝗬𝗏. Internal secretory cavities, they includes 𝗧𝗦𝗔𝗧 cavities and 𝗬𝗏𝗔𝗧 cavities. So first one, digestive glands. Rightly, digestive glands, characteristic feature of insectivorous plants or carnivorous plants. Insectivorous plants or carnivorous plants, They contains digestive glands. These digestive glands secrete proteolytic enzymes, means protein digestive enzymes like pepsin and trypsin. Then they digest that proteins into nitrogen. Then plants absorb that nitrogen. These are digestive glands. Second one, nectar glands. Nectar glands, characteristic feature of entomophilous plants or insectivorous plants. This nectar is commonly made up of sucrose, glucose or fructose. By this nectar, plants attract insects. This nectar is of two types. If nectar present on floral pods, rightly called floral nectarious. Floral nectarious, either nectar present on ovary or nectar present on stamens or nectar present on septa, rightly called septal nectarious. Ovary, example, Fabaceae members. Stamens, mustard, brassica. Septal nectarious, present in Liliaceae members. Other than floral pods, if nectar present on vegetative, rightly called extrafloral nectarious. Example for extrafloral nectarious, passiflora. In passiflora, stalk of leaf or petiole contains nectar. Then special inflorescence, cyathium. Special inflorescence in cyathium, outside the bracts, that a nectar is present. And all insectivorous plants in which that a modified leaves or that a trap leaves contains nectar. Third one, water secreting tissue. Water secreting tissue is called hydathode. Hydathode includes cavity, epithem, vein ending and water stoma or stomata. Actually, leaf tip or leaf margin contains large size spore. This large size spore is called water stoma or stomata. Below the water stomata, there is cavity. Below the cavity, there is colorless parenchyma. This colorless parenchyma is called epithem. Below the epithem, there is vein ending. This vein ending contains hydathode. It includes water stomata, below which there is cavity. Below the cavity there is colorless parenchyma called epitheme. Below the epitheme, veins. This veins contains tracheids. So generally, from tracheids, water enter into epitheme, then into cavity, then this water drops exude from this water stomata. Such exudation of water in the form of liquid through this water stomata is called guttation. Generally, root pressure is responsible for guttation. But a maximum root pressure, 1 or 2 atmosphere, so it explains ascent of sap only in herbs and shrubs. And this guttation, because of this reason, this guttation occur in smaller plants like grasses. And example for guttation, Colocasia, then 𝗧𝗧𝗧𝗃𝗃, tomato. This is about this external secretory cavities. Next one, internal secretory cavities. These internal secretory cavities are 2 types, 𝗧𝗦𝗔𝗧 and 𝗧𝗦𝗔𝗧. 𝗧𝗦𝗔𝗧 cavities. 𝗧𝗦𝗔𝗧 means these cavities are formed due to enlargement of intercellular spaces called 𝗧𝗦𝗔𝗧 cavity. This cavity is surrounded by a row of secretory cells called epithelium. Example for 𝗧𝗦𝗔𝗧 cavity, resin ducts in pinus. Next one, 𝗧𝗦𝗔𝗧 cavity. Cavity formed due to lysis or due to death of cells is called 𝗧𝗦𝗔𝗧 cavities. Best example for 𝗧𝗦𝗔𝗧 cavity, one is oil cavity in citrus, that is fruit wall pericarp. So these are special tissues, important internal secretory cavity, that is latissiferous tissue. This latissiferous tissue, it secretes a milky white substance. It is called latex. Rightly, latex is emulsion of proteins, carbohydrates, essential oils and resins. Then this latissiferous tissue found in plants which grow in tropical areas. And these latissiferous tissues are generally multinucleated. And this multinucleated condition in plants are called cenocytic. And this latissiferous tissue usually associated with complex tissue, that is what means phloem. Then this latissiferous tissue is of 2 types. One is latex cells and second one latex vessels. Latex cells are called non-articulated, and these latex cells may be branched or unbranched, but cytoplasm contains many nuclei, multinucleated. Then cells are living cells, and cell wall is made up of cellulose. Latex cells found in 𝗂𝗎𝗺𝗍 and also 𝗧𝗧𝗧𝗧. Example: 𝗂𝗎𝗺𝗍 and ficus. Next one, latex vessels. Latex vessels are called compound 𝗬𝗏 tissue or called articulated. Latex cells are called non-articulated. These latex vessels are branched. All these branches they fuse to form canal or channels through which that latex flows freely. These latex vessels found in families like 𝗂𝗽𝗱𝗁𝗂 and also Asteraceae. Example for latex vessels: hevea. Then importance of latex. In some plants, latex used in making rubber. So Indian rubber tree is a ficus elastica. Then para rubber tree, that hevea. In some plants, that a chicle, this chicle is obtained from latex of Acros sapota. It is used in making chewing gums. So here I'll discuss about some neat model questions. First question: multinucleated condition can be seen parenchyma, collenchyma, 𝗬𝗏 tissue and sclerenchyma. So answer is 𝗬𝗏 tissue. Why means parenchyma and collenchyma, they are uninucleated cells. Sclerenchyma is a dead tissue. Dead tissue means no protoplasm, means no nucleus. And answer 𝗬𝗏 tissue. 𝗬𝗏 tissue is 𝗧𝗏𝗏𝗃 tissue. In this, what condition leads to 𝗧𝗏𝗃? In this, only nuclear division takes place. Karyokinesis takes place. That karyokinesis is not followed by cytokinesis. Such multinucleated condition is called a 𝗧𝗏𝗃. Answer is 𝗬𝗏 tissue. Then second one, which of the following simple tissue shows maximum de-differentiation? Answer is maximum de-differentiation, parenchyma. Actually, differentiation dedifferentiation, redifferentiation. There are three types. Differentiation. Differentiation means that meristems, it gives cells like a primary xylem, primary phloem. So, these cells, for example, xylem is living cell. For conduction, it undergoes differentiation. How means? In that xylem initially cellulose is present, but after maturity lignin deposition takes place. For what means? Mechanical support. Then after maturity, that a protoplasm, it loses protoplasm. So, cells produced from meristems to perform functions, they undergoes differentiation. So, differentiated tissue, example, primary xylem and phloem. The next one, dedifferentiation. Here dedifferentiation means actual permanent cells, they lose their power of division either temporarily or permanently. So, there are some permanent cells, they regain their power of division and produce secondary tissues, rightly called dedifferentiation. Best example for dedifferentiation, medullary rays. This medullary rays, by dedifferentiation, produce interfascicular cambium. Dedifferentiation. Then another type, redifferentiation. Redifferentiation means secondary meristems, they produce secondary permanent tissues called redifferentiation. Here core cambium, it is secondary meristem, it produces secondary permanent tissue like a cork called a redifferentiation. So, differentiation three types: dedifferentiation, redifferentiation, first one generally differentiation. So, point is here medullary rays, they are parenchymatous. So, generally parenchyma shows that dedifferentiation. Another example for dedifferentiation, primary cortex. This primary cortex, by dedifferentiation, it gives cork cambium. So, even primary cortex is a parenchymatous. So, maximum dedifferentiated tissue is what means parenchyma. The next one, 𝗳𝗨𝗧. Once again I am telling, this is last year NEET question. 𝗳𝗨𝗧 means actually xylem plugs. Xylem plugs are called 𝗳𝗨𝗧. So, xylem means answer, heartwood. Rightly, secondary xylem is called a wood, but wood two types, heart and sapwood. Only vessels of heartwood blocked by these balloon-like structures, so called 𝗳𝗨𝗧. 𝗳𝗨𝗧, it shows both the disadvantages and advantages. Disadvantage, due to formation of 𝗳𝗨𝗧, that a conduction is stopped. That is why heartwood not useful for conduction. Advantage, this 𝗳𝗨𝗧 prevents that entry of harmful pathogens. 𝗳𝗨𝗧. The next one, P protein. This P protein present in cytoplasm of sieve tube. Actually, injured phloem is non-functional. Injured phloem This P protein blocks that sieve pores, so conduction get stopped. And tissue produced by, so answer for this P protein. So tissue produced by redifferentiation, just now I discussed. So answer is cork.